Okay.
Perfect. Thank you, Marcelo. Okay, my talk from today is related with those animals that live in the intertidal area. This is a summary of the different experiments and field surveys that we've been doing during the last 10 years, in not only in Chile, but in different places, New Zealand as well. So, and also this is part of my my actual project. That is a Fondesit grant, and this is the number of them. Well, my name is Víctor Cubillos. Everybody knows me as Mauro. I'm a marine biologist from the Universidad Austral de Chile, and I carry out my PhD at Otago University in New Zealand, where in 2011 I got my degree. Presently, I'm academic, I'm a scientist from the Universidad Austral de Chile, and I belong to the Institute of Marine and Limnological Sciences that belongs to the science faculty. My background is related to ecophysiology, and my research interests are also associated to reproductive strategies and reproductive aspects of different organisms, mostly in mollusks and sea anemones, and oxidative stress, cellular responses of animals to different environmental changes, and also photoprotection and photo damage in this kind of organisms. This is the science faculty of the university. We are in the first floor of this building. Well, why to study the intertidal zone, and what is the reason that push us to do experiments and do science in this area? It's because this is a really stressful environment. Animals are really exposed to a challenging environment where salinity, temperature, predation, drought, and air exposure can generate different kind of responses. Okay, so the intertidal zone has been mostly related to marine environments, but we also study the estuarine environments. We have in Chile different kind of estuaries, and some of them are quite big, and the other ones are really small, like 100 meters wide. So in this kind of environments, the sessile organisms can suffer a lot, and they have different strategies to make to address these environmental changes. So not only cellular strategies, but also behavioral strategies. Okay. One thing that we took advantage is that during late winter and early spring, the ozone layer gets thinner. So this is the reason, because a lot of ultraviolet B radiation penetrates through the troposphere and impact marine and terrestrial ecosystems. The ultraviolet B radiation is quite important because this 1% of radiation that penetrates through the troposphere generates more than 50% of photobiological and photochemical effects in marine organisms. This is a graph that we obtained from Rubin in 2005, and this is the daily cycle of ultraviolet B radiation, black dots, and PAR radiation as well. So you can see what is happening here in the mid-latitude of the south hemisphere, that at midday we have a really high peak. We reach the highest peak of the day. So animals can be exposed during low tide to this condition, can suffer a lot. I think everybody knows this curve is the tolerance curve, and we are working in this area, the stress zone areas, where few individuals can survive, and they have to display really efficient strategies to live in this environment, especially in the intertidal environments where the environmental conditions change according to the tide. Okay. This was the first field survey that we carried out during 2008, 9, and 10 in New Zealand. This was part of my PhD program, and I studied an intertidal community that was composed by two different invertebrates. You can see Dactynia tenebrosa, this red sea anemone that lives over the rocky substrates, and this other invertebrate that is a top shell. You can see clear difference between them. One has a physical protection but is conferred by the shell, and the other animal, the Dactynia, hasn't physical protection. So what we did with those animals was we surveyed them during 26 months in the Otago Bay. This place here in the South Island of New Zealand is like latitude 44, 45 degrees south. And we collect them during 26 months, and we analyze different kind of photoprotective compounds. In this case, I estimate mycosporine-like amino acids compounds using HPLC-MS, and we obtain this situation. This is this part of the graph. Sorry, this corresponds to the sea anemone. You can see increased levels of mycosporine-like amino acid through the time. That is not completely correlated with levels of ultraviolet B radiation. That is the upper part of the graph. You can see this is the annual cycle or biannual cycle of radiation at the study place. Okay. If we see what happened with the snail generate or acquire a reduced amount of mycosporine-like amino acids, but this less amount are shared between more different mycosporine than the sea anemone. Okay, so this was the first part of the study. Then we study the generation of mutations in the DNA. We collect animals from the period 2009-2010 in black bars and 2010-2011 in gray bars, and we compare the situation between the anemone and the snail through the time. This was the result. Again, the animal that lack of physical protection can acquire more mycosporine or more photoprotective compounds than the top shell, and that was correlated with the levels of CPD's concentration. This damage associated to the DNA, so this animal induced more damage than this one, again by the lack of physical protection. We also wanted to understand what was the levels of photooxidative damage in this organism, and we have a really nice tendency here. You can see with open dots the snail, that the snail generate elevated levels of lipid peroxidation, especially during summer seasons, and then get reduced during autumn, winter, spring, and rise again to the summer. So if we see what happened with the sea anemone, the sea anemone reduced its levels at 60% lower than the top shell. So these animals is better, can tolerate in a better way the environmental fluctuation that happens in the Otago Peninsula. So protein carbonyls have in the same situation. You can see elevated levels, similar levels between summer and winter. Between the top shell and the sea anemone, and then the levels get reduced like 80% of what's happening in summer. So we have a clear picture that although this animal has a lack of a physical protection, they have strategies that allows them to live in an environment that is really hard. Both animals are intertidal animals, are exposed what happened, we also follow some antioxidant enzymes, superoxide and catalase. We see what is the situation. The animal, the top shell has increased levels of superoxide through the year. The same situation happened with catalase here. So the sea anemone can manage how they address different kind of stressors in the environment through the year, especially during summer and the cold events during winter. Sometimes in this area, the temperature drops under zero, so the animal is quite can manage the situation quite well. The only difference is that the sea anemone, in this case, the glutathione reductase, can increase, or their values are higher than those observed in the, so the GPOX activity is higher in the anemone than in the top shell situation that explain clearly that the detoxification mechanisms can be more efficient in this animal than in the top shell. Later, we carry out a control experiment. We finished this field survey, and we decide to understand how anemones respond to different scenarios of radiation and how temporal acclimation influence the cellular response of the animals. So we collect sea anemones from that were acclimated to summer conditions and winter conditions as well. And we expose for 4 days to the different radiation treatments. We expose animals to only PAR radiation, that is that includes wavelengths between 400 and 700 nanometers. Then we generate a combination of PAR and UVA. So the spectrum of radiation involves radiation between 320 and 700. And then we include B radiation, and also we create a special B treatment with a 20% increase over the normal values, simulating an ozone depletion condition. So we expose these animals inside of this growth chamber, and the idea was to understand different things. The first thing was associated to the direct DNA damage. So ultraviolet B radiation can generate damage, but what happened here is In summer acclimated organisms, the CPDS doesn't change pretty much through the different treatments. However, in winter acclimated organisms, we can see a significant increase, especially in those treatments that include ultraviolet B radiation. So there is a temporal effect that generate different responses in animals. So animals that were acclimated to low levels of radiation and are suddenly exposed to elevated levels increase those levels of DNA mutations, okay? This is the mutation that is generated in the DNA by the absorption of UVB radiation. The photon can generate a covalent bond between thymine, so generate a nick in the DNA strand. So that generate the mutation and has problems during the replication and transcription of the information. The other thing that we measure was how these animals respond in terms of oxidative damage. We saw that summer acclimated organisms increase their protein carbonyls and also lipid peroxide, especially when they are exposed to the full treatment of radiation, but also increase in a higher percent when the level of ultraviolet B exceed 20% more the normal values. What happened in winter acclimated organisms? They are less, they are more susceptible to the radiation, so they increase even more their levels of protein damage and lipid peroxidation levels than summer acclimated. So again, the temporal is not the same. Collect animals from winter and summer and expose them and do the experiments in under controlled conditions. You have to consider that animals have responses and they are acclimated to different situations, okay? That is given by a temporal gradient. We estimate also superoxide, catalase. We see different tendencies, and most of them are associated to increases in ultraviolet B radiation. The B radiation generate more problem than A and also PAR. So you can see the tendencies there. Same with GPOX and GR. We estimate also glutathione with different tendencies, most of them the same, increase especially during the B treatments, okay? We here we shift to another animal. We carry out different experiments. This organism is Crepipatella dilatata. This organism is estuarine organism, is a sleeper limpet that lives attached to harsh substrates, generally shells or rocks that are in the bottom of the estuary. And these organisms is a really good model because has something that is really special. The reproductive mechanism of these organisms involves the change of sex through the time. So they have changes of sex during the ontogenetic development. So for example, in the picture, you can see these small organisms. This organism is a male, and the male start to develop and increase in size, and then start to appear the female's organs, and then turns into a female at the end of their lives. So the animal at this moment can lay the eggs over the substrate, and the animal broods the capsules under the shell in the pallial cavity of the animal. So what's happened with the animal? The animal is over the rocks or over the hard substrate. The animal the female open the pallial cavity, allow that water comes into the pallial cavity. The animal can filter the water, can obtain food, and also generate the oxygenation of the pallial cavity where the babies are located. Okay? So what we did in this experiment, the idea was to understand how salinity changes can generate the isolation of the animal from the environment, and also can reconnect the animal with the environment when the salinity goes up. So this is the situation. We generate this scheme. For example, we play with the salinity here. We use water with 32 PSU of salinity, and that allow us to get the animal connected with the environment. So the animal was open and generate the flow of water inside the pallial cavity. Then we reduce the salinity to 10 PSU. So the animal generate the isolation. The animal close, contract the muscle of the foot and get close, so it isolate themselves from the environment, and the animal fell into hypoxia and then an anoxia. And this situation, we project this situation during almost 72 hours. Why 72 hours? Is because in this place, in the Campian estuary, the water, the estuary is so small that when rains, like 2 or 3 days or 5 days, the salinity can get reduced to 7 PSU during 4 or 5 days. So the animal get isolated during that period of time from the surrounding environment. So the idea was to understand what happens later when the animal get reconnected to the environment. So in this moment, we increase the salinity, and the animal has to pay the oxygen debt, because the animal fell into these two conditions, hypoxia and anoxia. And then when the pallial cavity reconnect with the environment, they start breathing again, So this is the moment when the animal generate a problem here. So we took females brooding different developmental stages of their embryos, their encapsulated embryos. So we have females brooding early stages, female brooding belliger stages, and female brooding pre-hatching stages, and also embryos by themselves, So we estimate lipid peroxidation, and you can see here when you generate the isolation of the babies, nothing happens. Just increase a little bit of lipid peroxides during the isolation. But when you reopen, or when the mother reconnect again with the environment, and the oxygen flows inside the pallial cavity, the animal babies increase their oxidative damage level. Okay, same happened with the adults. You can see here, adults brooding early stages, belliger stages, and pre-hatching stages. Oops, sorry. Here, the same with protein carbonyls. We have the same situation after or during the reoxygenation of the pallial cavity increase oxidative damage to the proteins. You can see the same tendency in all of them. There is this case, there is a 30% more or less of difference between the other two developmental stages, probably increased by a differentiated oxygen consumption rates. We also estimate superoxide, and these enzymes increase their levels later in response of the levels of lipid peroxidation and oxidative damage to the cell. We also estimate different, I didn't want to put all the results, but we also estimate catalase, glutathione, glyoxylase 1, glyoxylase 2, and all the levels increase between the hour 84 and 96. So they have a really nice tendency to increase in response of the oxidative damage levels. After that, we discovered this little sea anemone. Its name is Anthopleura hermaphroditica. This animal is a Cnidarian, is a relative of the corals. Its latitudinal distribution in Chile ranged between the 23 degrees to the 41 degrees south. They live in intertidal and subtidal areas of bays and estuaries. But the interesting thing is that they live buried in the sediment. So they project only the tentacular crown, where the small symbiotic dinoflagellates are located. Okay, so therefore this animal is a mixotrophic organism, and they also use as a reproductive strategy the internal brooding of embryos. So the adult can brood embryos between 1 and 15 or 20 embryos simultaneously. So this is a really interesting model for us. So what we did with this animal, the first thing was generate a characterization of the environment. The objective was to understand how low tide that happens at midday, when the radiation, the ultraviolet B radiation, reach the maximum peak, influence the photooxidative response of these organisms acclimated to summer and winter environmental conditions. So we carry out some collection during summer and during winter, especially during clear sky day, and we start collecting animals at every 2 hours from 8 a.m. until 8 p.m. Okay, we estimate also ultraviolet B radiation and PAR radiation. We use a portable radiometer to obtain that information, and salinity and temperature. What happened here? This is the salinity during summer in black bars. You can see the summer situation, and this is winter. The salinity get reduced at midday and reach again initial values. So this is the low tide condition. What happened with the temperature during summer? The temperature increased from 14.8 Celsius degrees until 31 or 32 degrees. So the animal are exposed to a thermal condition that is really hard, while during winter is pretty much stable. Ultraviolet B radiation increased to really high levels. We can obtain almost 2.3 watts per square meter. during summer, and during winter the level is really low. So during summer we have a really nice component of damage here that is induced by ultraviolet B radiation, and also we have levels of PAR radiation that reach almost 2000 microeinstein per square meter second. Okay, so this is the situation during summer. What's happened here in terms of oxidative damage? We have lipid peroxides levels that pretty much does not change. Statistically, it does not change through the time, but during summer we have 2 or 3 times higher levels than during winter. Same situation with protein carbonyls. We obtain almost 6 times differences between summer and winter. We also carry out a principal component analysis to understand the data, and a good things happen here because pretty much temperature and ultraviolet B radiation are responsible of most of the damage generated during summer, while during winter the changes associated to salinity are associated to generate more damage in the animal. So this animal lives in an estuary. The estuary generate daily conditions that stress animals, but also the animal are exposed during all the year to different conditions that implies that they are continually exposed to a stressful condition. So the animal has to have really good mechanisms to avoid the damage or tolerate the damage. This is the reason, because the animal continues living in this area. Something happens in this area that is good for them. Otherwise, the animal will disappear. Okay, so we estimate the total antioxidant capacity. We saw that during winter, animals increase their levels, and we associated these levels to an increase in phenolic compounds that also generate antioxidant response in animals. So we carry out this experiment during 2018. We characterize the situation. Then we generate some sort of different experiment. This is a multifactorial experiment. We play a little bit with salinity, ultraviolet B radiation, photosynthesis, and oxygen consumption rate. This was a really nice experiment. That took a lot of time. We estimate we put the animals in special chambers. These are respiration chambers, but are not ordinary bottles. These bottles are made of quartz, so this allows us to expose them to different radiation conditions. Okay, we put the animals in the respiration chambers. We generate different radiation conditions using filters. Okay, and we exclude the photosynthesis using DCMU. That is a chemical compound, and we compare the situation between adults and juveniles exposed to different salinity conditions. So we have a multifactorial experiments here. Pretty much the results are these. Animals exposed to 30 ppt or part per thousand are pretty much open. They are really.
Mauro, can you explain the logic of DCMU before?
Sorry, Marcelo. See, Yeah. Okay. DCMU is a chemical compound that generate inhibition of photosynthesis. So if you add DCMU to the water, you generate the inhibition of the photosynthetic process, and these small anemone are symbiotic organism, so they also generate oxygen by photosynthesis. So the idea was understand how these animals get impacted by different levels of salinity and radiation. The logic of the basis of this experiment is because most of the experiments that we carry out in ecophysiology using mollusks, for example, we use the oxygen consumption rate as an indicator of physiological stress. So when animals are stressed by a different environmental factor, the oxygen consumption rate gets heavily impacted. So in this case, as animals, as the sea anemones has symbiotic dinoflagellates in the tissues, we need to block the photosynthesis and in that way understand what is really happening with the animal. Okay. So after that, animals that were exposed to 30 ppt show a really nice behavior. They are open, the tentacular crown are completely open. But when you expose the animals to different, to reduced levels of salinity between 22.5 and 15 ppt, they start to reduce the exposure surface of the animals. They start to generate a contraction of the column, and after that, more reduced salinity generate that animals get in. So the tentacles get in the animal and generate the maximum compression that reduce its surface. It exchange surface with the environment. Okay, so what we see here, that in terms of oxygen consumption rates, animals with that salinity, but while you reduce the salinity, the oxygen consumption rate get lower, and you also can see in this case that DCMU, the use of DCMU can show you how is the real consumption of the animal in terms of oxygen inside of the chamber. And in that case, we have negative values because juveniles animals has the zooxanthella or the symbiotic microalgae of these juveniles generate more oxygen than they can use inside of the chamber. So you can also see a reduction in the production of oxygen inside of the chamber. In terms of oxidative damage, we have a really clear tendencies. DCMU animals without DCMU generate increased levels of damage, and this damage also increase when you reduce the salinity. So when the tide is coming down or animals are exposed to reduced salinity in the estuary during a long period of time, can heavily increase levels of damage. And the same situation happen with juveniles. So we have clear tendencies there. Total antioxidant capacity is not it's not really clear. we have more constant levels. Probably the animal use different strategies to address these environmental changes. Okay. Then we carry out another multifactorial field survey. That the idea was understand how plastic this animal is and how the local adaptation defines the response of these animals in these small estuaries. So what we did here, we took animals from this area, subtidal animals and intertidal animals of this area. We compare both situations that were apart like 4 or 5 meters, and we saw difference. This is the experimental setup. We use also radiation. We use different salinities, different temperatures, and we follow the animals through the time. So we have this. This was published in PLOS ONE during January of this year. We have that intertidal organisms sometimes respond better than subtidal animals when they were exposed to different experimental conditions. You can see here temperature. Intertidal animals tolerate better the temperature than subtidal animals. Same for salinity and the other radiation factors. Protein carbonyls is not pretty clear, but some tendencies can be observed. However, the antioxidant capacity shows us that intertidal animals respond better than subtidal animals when they are exposed to radiation, temperature, and salinity in this estuary. We carry out some specific experiments with, to understand why this animal lives buried into the sediment. So we carry out a behavioral experiment and also a cellular response experiment. So we expose animals in the behavioral experiment. We put them over the surface of the sediment. We turn on the lights and we calculate the time and the depth and the velocity of the animal that took them to get buried inside the sediment. And the results are here. We have the situation is the follow. The animals that this is the adults in black bars and the juveniles in white bars. So we expose them to different radiation conditions, and we see that animals that are exposed to ultraviolet B radiation, they accelerate the movement of the body column, and they get buried faster than animals that were exposed without ultraviolet radiation. That, and also we have here levels of protein carbonyls. We have adults are less susceptible to the radiation than animals that are in a lower developmental stage. So juveniles are pretty much susceptible to the damage than adults. We have some difference here in antioxidant capacity as well. Then we've been.
Can you explain the difference between black and white in this one?
Yeah. Well, this is the experiment with sediment and without sediment. This is the second part of the experiment. So we expose the animals with sediment to the radiation and without sediment. So the idea was understand what is the role of the sediment in the life of this animal. If it represent a physical protection to the animal. So animal with white bars represent animals with sediment, okay, and without sediment with black bars. So animals with white bars has less antioxidant capacity than animals that were exposed without sediment to the radiation. You can see some slides slightly different, but sometimes it's not completely you cannot see a completely clear difference between them, okay? But in this case, juveniles has different situation, different conditions. So the animals that were exposed to ultraviolet radiation with sediment generate less damage than animals that were exposed without sediment. Okay, so sediment represent a really good physical protection in animals that lack of physical protection by themselves, like a shell or some hard cover that protect its soft tissues. We've been there is nothing. There is no information associated to the brooding aspects of these organisms. We've been working with that. Our team carry out different histological cuts of animals, brooding animals. You can see here, this is a transversal cut of the animal. This is the pharynx of the animal, the mouth of the animal. And you can see here small babies inside the mom. You can see one embryo and two embryos inside of the gastrovascular cavity. Here we have a juvenile. This is a big juvenile that is inside the mom's body. A small planular larvae that is inside also of the gastrovascular cavity. We characterize with electronic microscopy the different developmental stages. There is one missing stages, that is the planular larvae that we are working now. We estimate also the percentage of brooding animals through the different seasons, number of adults that brood, and also the number of brood embryos inside of different adults. Okay, and then we ask, we generate this question if the brooding is a protective mechanism for embryos during the salinity changes in the estuary, and we expose animals containing embryos to salinity reduction during 72 hours. And we compare the situation with embryos that were released from the parents. So we obtain this information. We have a lipid peroxidation here. So incubated embryos inside of the body of the adult get less damage than those that lack of physical protection of the parents. The same from protein carbonyls. We have the same situation, and the antioxidant capacity is 50% less than those animals that were that lack of physical protection. So we understand that the parents protect embryos from different factors The first one is the ultraviolet B radiation. So generate this condition that allows animals to live into the sediment, protect physically to ultraviolet B radiation, and also the parental care generate a physical protection that is generated by the adult. So brooding is a mechanism, is a reproductive mechanism that is really common in this kind of organisms, especially in estuarine organisms. So, for example, we have the Chilean oyster, we have Crepipatella dilatata, that is this snail that also incubate their embryos, and we have this little anemone that lives in the same estuary. So we have 3 different models that present the same reproductive mechanism. So the idea is the response is the same. Animals that live in a hard environments must protect their babies from the environmental conditions that is really fluctuating outside. Okay. We've been working with immune response. This is a work that is carried out by a thesis student, undergrad thesis student. We are carrying out some experiments associated with the reinfestation. So is the contrary process of bleaching. You know that when these animals are exposed continuously to different stressful conditions, they release their symbiotic organisms. So in this experiment, we need to pass from this condition, the A condition, to the B. So the idea is to follow in the time how long takes this animal pass from this condition to this other one. This is something that we already start a couple of months ago. It took a lot of time to obtain these animals, pretty much 2 months. We've been doing this simulating tidal cycles and see what's happening with the levels of oxidative damage to the lipids and the antioxidant capacity of animals. This was part of an undergrad thesis of our lab, and we use these images, these pictures, to quantify the mitotic index. This is an image that was taken using a fluorescent microscope, so you can see the division of the cells. You can estimate. This is a physiological parameter to understand how is the condition of the animals in terms of stress. So when animals are in a condition that is optimum, in the physiological optimum, the percentage of mitotic index is associated between 20 and 24%. So knowing that, if we expose animals to different salinity conditions, we can observe that the mitotic index get reduced in time. Okay, or pretty much animals reduce the mitotic index to a 5%, 10%, more or less, while animals exposed to 30 PSU can reach almost 20%. So 20%, 30 PSU is the salinity where animals lives in a condition, in a physiological optimum condition. If you reduce that salinity less than 22.5 or 23 PSU, the animals start generating problems. So this is the reason why we use that salinity as a mark. Okay? We've been also studying how this animal regulate this cellular volume in terms of salinity. This electronic microscopy image where it show the different zoosantella. This is the small dinoflagellate that lives inside of this symbiotic capsule in the inner part of the animal. This is the gastrodermis, the inner part of the the sea anemone, the tissues of the sea anemone. The idea was to estimate how salinity can generate damage to the membrane of the cell, and how is the level of destruction of the chloroplast inside. So we carry out experiments, and after this talk, we need to go to the electronic microscopy unit to see our cuts, to see what is happening with that. And I would like to finish my talk showing some progress, some work that we've been doing with other colleagues here in the Institute, and we are working with this small bivalve. It's a mussel. It's a small mussel that measure like 4 or 5 centimeters long. And what we did here, we carry out a field survey. We estimate oxidative damage between animals from the upper intertidal zone and the lower intertidal zone through the clear sky day in Valdivia. And we also generate a transplant experiment. We move those from the lower to the upper part and the upper to the lower part. So the idea was estimate different kind of cellular markers. And also we carry out a field survey during November last year with a colleague, Edgardo Cruces, and our team. The idea was understand what is happening with this sea anemone. This is Antopleura, is the symbiotic anemone that lives in the estuary, in terms of photophysiological parameters. So he brings to the field his equipments to measure this kind of parameters. This is the electron transfer rates, the different fluorescence parameters associated to photosynthesis. And the idea was to understand, okay, what is happening during the night? Because we have the idea that animals can generate damage during the day, but what is happening during the night? So this is the reason, because we carry out this 24-hour cycle in the field, how they recover from UV and some oxidative damage and cell markers. Okay, and what is next? The idea is to work with this sort of animals. There is a really complex problem here in Chile, because Metridium senile is an invasive sea anemone that is generating problems, especially with some other aquatic resources like the sea urchins. So they are expanding fastly, really fast in the system. But the situations imply that the sea urchins are moved by the presence of this organism. This is another one, Diadumene lineata, appears in Chile a couple of years ago. And the idea is to understand what is the cellular response of these animals in relation with this. This is Phimactis. This is a native species, and how they will behave in the presence of each other, and how how is the species that are going to win. So this is the situation, and this is the final of my presentation. Thank you, Marcelo.
Oh, thank you, Mauro. Thank you. Thank you. Thank you very much. It's very, very good. Very good. Yeah. So
It's
I'd
a
like
lot
to
of
open.
it's a
Yeah.
lot of information.
Yeah.
Sometimes took a lot of time to explain some things, but sorry for my English. It's not the best, but I will do my best.
Oh, your English is very good. No problem. So it's really a lot of results. So the people that enjoy marine biology had a very good overview of your work on free radicals, antioxidants, UV radiation, salinity. So it's really impressive. I'd like to open for the other people to ask questions. I will have some at the end. We are starting work with these organisms, so we're really interested here. But please make questions. Opening for the others. So while people is thinking about questions, I'd like you to explain more on the effect of salinity. Why higher the lowest salinity causes more oxidative stress?
Of animals has these animals, the sea anemones, are osmoconformer organisms, so they have to change they have to reach an isosmotic balance with environment. So that situation generates some osmotic changes. So this is the reason, because low salinity generates oxidative damage. We saw we've been doing different kind of physiological experiments, not with anemones, but with oyster, the Chilean oyster, and we understood that when salinity drops from 22 or 22.5, the animal get close. We use different kind of electrodes and different kind of sensors that we put through the shell of the oyster, and we estimate that we compare the salinity of inside of the pallial cavity and outside of the pallial cavity, and we obtain information that the salinity of inside was always over 22 PSU. So the animal physiologically responds to this situation, getting closing its valves. The animal close its valves, get isolated from the environment, and this water remains inside of the pallial cavity. So one of the problems that happen here is, okay, you don't know, or the animal doesn't know how long this situation is going to be projected in time, because if project too much, the animal start to producing waste compounds that are released into the pallial cavity. So the animal get intoxicated in some point. So they have to reduce their metabolism to avoid the toxicity that the animal can generate by itself. Also, the situation of oxygen, the oxygen consumption. The oxygen is a factor that is limited inside. So if you get isolated from the environment, you have a certain amount of oxygen available to breathe during a period of time. So you have to use this resource really well planned in time. So this is the reason, because the animals fell into hypoxia, anoxia, and then when they get reconnected again with the environment, it opens and start breathing again. And this is the problem that increase the levels of oxidative damage. And this is the situation that we saw, and for example, in Crevipatella. Crevipatella generate this oxygen, the payment of the oxygen debt. There is a debt of oxygen consumption that is generated by the isolation of the animal with the environment.
Oh, yes. These results with Quiribatella, I thought these results are really nice because you see the animal going into hypoxia, anoxia, and then reoxygenation. And what I thought interesting is that you did see a small increase in oxidative stress under hypoxia, because it's counterintuitive. I mean, 10 years ago, people would not understand that with low oxygen, you get more damage. But your experiment it's incredible because you see a small increase in oxidative damage in hypoxia. Then when you have reoxygenation, boom, and it goes really high in lipid peroxidation and carbonyl protein. These data it's a kind of textbook data for everyone. It's perfect. I never seen results like this in our area, comparative biology, only in rats. Those models of ischemia and reperfusion. Yeah, it's really, really nice. And you did that in several conditions. So it's okay, it's good. So see if other people have more questions. Oh, okay. I have one more comment. When you show your studies comparing, let me see, winter versus summer in the anemone,
Okay.
it was, and you compare several different times of the day, and it's really incredible the difference between radiation in summer versus winter. I mean, you're not in the photochem. I mean, environmentally, how can you explain why it's so different, the radiation between summer and winter? We understand, of course, temperature is different, but why radiation is so hugely different?
Well, the first thing is every season generate a different distance of the sun with the surface of the earth. So this is the one of the main reasons, and also the angle of the sun respect to an object change the distance. So this is the reason, because at midday you can find the maximum daily peak of radiation. This is the reason because, for example, during summer, you have levels, the highest level of the day between 2 p.m. and 3 p.m.
Yeah, it's because you're very much in the south. Yeah, if you go
exactly. We are really close to the mid latitude. So the mid latitude is almost the closest place during summer. So also the radiation, the ozone hole, generate the penetration of different wavelengths, and especially ultraviolet B radiation, not in a perpendicular way, but also in that way as well. So all the south cone of South America is impacted by this situation. And this is the reason, one of the reasons, because we carry out this experiment with ultraviolet B radiation. We have the lamps, we have the filters, we have the radiometers and everything, because here in Chile is a problem. It's a problem.
Yeah, especially in the south of Chile. Yeah.
So exactly.
Yeah.
Because you have if you get closer to Antarctica, you have more hours of sun. So, for example, here in mid latitude, we have almost 18 hours of sun during summer. And if you go to Punta Arenas, this is the south part of Chile, you have the sun fell down like 11, close to 11:30 p.m. So you have sun in a long part of the day. So the accumulation of the damage in animals is something that is really impressive, how they accumulate damage and how they get recovered as well. So understanding these strategies is
the difference in damage between summer and winter is very high. And the other interesting thing is the antioxidant capacity. The result is the other way around, because in summer they're using this antioxidant capacity to protect. So that's
Exactly.
why it's low. Yeah.
Exactly. Exactly.
Yeah.
And they use another strategy as well, because these animals, the sea anemones, use mycosporine-like amino acid as well. So they use a photoprotective mechanism. We didn't calculate, for example, the carotenoids, but they have carotenoids. They have mycosporine-like amino acid. They have really good strategies, for example, related with heat shock proteins and blah, blah, blah. So the animals have a sort of different strategies to deal with these changes in the environmental conditions that is get by tidal reduction and also the natural daily cycle of radiation. So
probably heat shock proteins is probably hugely different between summer and winter.
Yeah, sure. That is one thing that we would like to point in this next steps is try to get associated with someone that help us with this situation. We are not geneticists, but the idea is to establish collaboration with someone that could help us to understand these aspects of the animals.
Yeah, that's the whole idea of this club, for people can collaborate. Yeah. It's very good. So now I'm passing for others to make questions. Otherwise, I will monopolize you. Seems that's not, people are not asking questions. There is a question. It's no, I thought there was a question in the chat box, but it's not. So seems that nobody is asking questions. So I would like to thank you again, Mauro or Mauricio.
Thank you, Marcelo. Mauro is okay.
Yeah. So it's very, very nice. So we of course, we are interested in cnidarians now. We keep changing animals. This kind of work we do here. But now we've been interested in marine biology. It's so we will be talking a lot in the near future. And I would like to thank everybody that has attended the seminar. It was really good. So a lot of basic results. And we would like to invite you for the next one. We're contacting Professor Niu from China to see if he presents his work, very nice work on Tibetan frogs. They live at very high altitude, like almost 5,000 meters, and compare with others not so high, like 2,000, and they saw incredible differences between these two groups of animals. So thank you. So I'd like everybody to open the camera so we can have a picture. Everybody, yeah. people can open the camera so we can, record everybody. Have a print screen. Yeah. Okay. So thank you again.
Thank you very much.
See you guys in the next meeting.
Okay. Thank you, Marcelo.
Bye-bye.
See you.
Thank you.
Thank you.
Much. Bye bye.
Bye-bye.
See you soon.
See you.
Bye.